Yes.
I may not have the right terminology here, but is it possible to artificially produce an osmotic membrane?
What? An osmotic membrane?
Yeah.
Yes, it is. And so cellophane will work quite nicely, certain kinds of cellophane. And this is kind of fun. You can take a tube, chemists call a thistle tube, it's just a long glass tube with a funnel in the end. Well, it's a glass funnel, I guess. But it sort of looks like a thistle. Usually you hold it up like this, and you pour things in it. But you can take a thistle tube, and it's got a little, it's round, and it comes in a little dent and a little bulb and a long tube. You can put a cellophane membrane over that and just strap it on with a rubber band. Well, before you do that, put your finger over the end of it and pour some syrup, in this thistle tube. And then, I guess somebody else helps you put the membrane on while you're holding the end of it like this, strap it on, and then turn it upside down. And you've got this, in the right kind of cellophane, won't let the syrup out. And so it's an osmotic membrane. The holes are big enough to let water through, but they won't let the sugar through. So you put this in a beaker of water, and the water begins to flow in. It flows from 100% water, to 80% water. Water's flowing from high to low concentration, and the sugar molecules are blocking the hole so it can't get back out again. And so the water begins to move up the tube. And so you can watch, it's a common lab experiment, and I just love doing these things. And you can just see the water go right up the tube. And sometimes, you can get it to actually make that fountain effect, just like you could with a plant. Although in this case, it's just an inanimate membrane. If you want to do it better, it's a little hard on the frog. Okay? A frog skin used as a membrane for an osmometer. I had one of those things that virtually shot water like an artesian well, and so they can really do it. Now it was still partly alive, at that point. But you can't have inanimate. Of course, there are water softeners, aren't there, that work on reverse osmosis and things like that. Osmosis is used to separate water and salts, so it's a common phenomenon that applies to living and non-living things. By the way, if there's sufficient demand, I could give you one more plant example that I was going to include but forgot.
Yeah.
All right, okay. That was sufficient. All I needed was one. Let's see if I can find a sheet to write on.
It's cold outside.
Yeah, no point going outside. By the way, I should know this, but do we have refreshments?
Yes.
Oh, okay. In that case, I'll make this a little quicker. I was just going to try to find something I didn't mind marking up. And maybe I'll find something I do mind marking up. Because did Mark ever get back? We went out looking for some magic markers that weren't permanent.
Couldn't find any.
Okay, all right. I can mark this one up. Okay. Is that one that doesn't erasable? We've tried it, right? Okay. Here's one of the most phenomenal. I think this may be a dry erase. We'll see if this works. Okay, picture. Here you've got the surface of a leaf, and there are little breathing holes in leaves. Called stomata, are these breathing holes. And if you look at one of these close up in a leaf, you'll find that there's a couple of kidney bean shaped cells like this. I'll draw it open like this. These are called guard cells. And they have a thick wall. This is just an ordinary wood wall, a thick wood wall toward the inside, toward the opening. So on low power, they kind of look like dark round circles. And then they're embedded in some epidermal cells in the leaf, just the outer layer of cells in the leaf. I'll surround guard cells like that. So this would be the guard cell. And this would be the stomata, the opening. Now, there's a couple of things to notice. The guard cells have much thicker walls in general than the just regular skin cells of the leaf, the epidermal cells. And they have a really thick wall toward the opening. And these things can close like this, or they can bow open like that. So they open and close like this to let air in and out and leave. Well, also in the guard cells are chloroplasts. Well, that turns out to be kind of crucial. Chloroplasts are the green things in plants that carry on photosynthesis. And they're in the guard cells, but they're not in the leaves, or not in the cells around the guard cells. The other leaf cells don't have them. Well, that turns out to be kind of crucial. And so this would be a guard cell pair closed. We'll just have this thick wall, next to the thick wall of the other one, like so. And then here would be the epidermal cells. Well, this might be the situation, we'll say on average, this would be a situation in night. And so the plant is not really, actively photosynthesizing. It's just sleeping. It doesn't really need to take in a lot of carbon dioxide or anything. Now, this is just an average. And then in the daytime, it opens up and takes in carbon dioxide to make sugar and so on. And, of course, uses some oxygen overnight. Well, how does it do that? How does that open up? Well, this turns out to be fairly phenomenal. And so, what happens here? It is closed like this. Well, at sunrise, the sun stimulates photosynthesis to begin in these are green cells, the other are clear cells. So in these green cells with the chloroplast, photosynthesis begins. Well, photosynthesis absorbs carbon dioxide, converts it into sugar. And so carbon dioxide is being used up inside the guard cell, but not in the surrounding cells, because they don't have any chloroplast in them. And so as the carbon dioxide is used up, that carbon dioxide is somewhat acid. And so as that's being used up, the cell gets more basic, less acid than the cells around it. Well, that activates an enzyme. And so enzymes or proteins are all twisted around in funny shapes. And the shape depends on how hot it is, how many salts are dissolved in it, and especially what the pH is, whether it's acid or base. So in one position, when it's mostly acid, the protein be coiled like that. As the carbon dioxide is used up and gets basic, it changes shape. And when it changes shape, it assumes a shape that can digest starch and break big long chains of starch down into separate sugar molecules. And so as this enzyme breaks starch down into sugar, the sugar molecules begin to fill up the cell and raise the osmotic pressure. So one big starch molecule has a lot less osmotic pressure, does a lot smaller job of blocking the exit holes than lots of smaller but still big sugar molecules. So changing starch into sugar raises the osmotic pressure. And so as the osmotic pressure is raised, well, the cell wall stretches, that'd be Turner pressure, but the cell walls are unequal in thickness. And so they're just thick where they touch each other. And so as a result, as the cells begin to swell, they change shape, they bow outward. One way you can demonstrate this to students is to take a balloon, just kind of a sausage-shaped balloon, blow it up, part way, and put a piece of duct tape. And as Mark knows, duct tape is one of my favorite things. So put a piece of duct tape on the inside. And in fact, if you want to make a better demonstration, take two sausage-shaped balloons, put duct tape here and duct tape there, and then tie them together. Or tie them together at the top, leave a little, at the bottom you can blow air into them. And as you stretch, they'll bend more on the outside, so the inside curves open. So you can make those balloons open up. Well, the extra osmotic pressure causes the cells to swell, but they swell unequally because of this unequal thickening of the wall. The stomata opens up. Okay. Now, who gets the glory for that? Okay, is that a result of random chaotic motion? Well, no. It certainly used random chaotic motion. This is another example of osmosis, of water molecules diffusing from a high concentration to a low concentration at a definite rate in a definite direction to a definite endpoint. And yet it accomplishes something absolutely marvelous. But it wouldn't work, for instance, if the other epidermal cells also had chloroplast. They would swell up and they'd push the guard cell closed. And so you have to design a genetic system that instructs the plant to make leaves where only the guard cells have chloroplast. Now that's more than one gene it would take to specify that information. And then you have to say, by the way, I want you to make cells that have a thicker wall where they touch than where they don't touch. That takes a lot of genetic information. And that's just awesome, you know. And by the way, there are no, you know, land plants, no plants that, you know, live in the air that don't have these. And so anything that requires this, they've all got them. And the ones you find in the fossils and the very first plants buried in the flood, what the evolutionists would say the first plants ever to evolve, have all this. All of these things are the same in the first, quote, simple land plants as there are in the most advanced plants they have today. Right there from the beginning, created, you know, right from the word go. And so now what happens in the evening? Well, in the evening, okay, the photosynthesis dies down. So the carbon dioxide is not used, it builds up, allows the cell to become more acid, which takes that enzyme, goes unfolded, crinkles it up, quits breaking down sugar. Another enzyme puts the sugar back together to form starch. The osmotic pressure is reduced. The snap of the cell walls draws them back together again, and they close. Wow. So tomorrow, okay, some of you have blades of grass showing already. And if you do, you know, you see sun shining on a blade of grass. This is happening millions of times over in the average leaf or blade of grass. Wow! You know, each one opening its mouth in praise to its creator as the sun comes up. And the design, what do evolutionists say about this? Absolutely nothing. I've never heard or read or even heard anybody talk about saying anything about what they all describe them. You know, they all list them. They all show pictures of them. You know, in fossil plants and modern plants. And I've had a lot of evolution. I think most of you know. I used to be a very, very enthusiastic, you know, evolutionist. Dr. Gish used to say I knew more evolution than any evolutionist debater he'd ever faced. And I never heard anything about this. By the way, what do evolutionists say about how plants get watered at the top of the redwoods? They just give you the numbers. Say nothing about it, miss the whole point, you know. And it's funny, you know, you can read my biology class, and well, I got a little booklet on this, you know, in a regular class we say the same things. But boy, oh boy, there's so much more to say. And, you know, sometimes, you know, by the way, just as a little political parenthesis, when did science scores begin to plummet in America? You know, America used to be a leader in producing scientists for the world, and scores began to plummet right after, who knows?
Prayer out of the school.
What was that?
Prayer out of the schools.
Well, it was actually a little before that, but prayer out of the schools didn't help any. Well, you know, when the Russians beat us into space, the government said, you know, that can't happen. We got to make every American kid a scientist. And so they produced government-sponsored biology textbooks, the BSCS blue, green, and yellow versions. And then so now they wanted to have a theme. You know, what theme are we going to use? I know, evolution. And that's when they quit teaching evolution in schools and started assuming evolution. No longer brought it up for questions. It wasn't fair to talk about. He assumed evolution was true. And as that, I was just beginning my college teaching career then, if this gives you some clue as to when this happened. And we were gearing up for the hot new students we'd be getting out of these super, you know, high school biology classrooms. 2 years later, we were doing remedial programs, you know, on how to do basic biology. And the score, and you can chart it, I mean, it's just recorded. The scores dropped down. Japanese, Chinese, just begin to plummet. Where are we? 7th or 8th in the industrialized world or something? Maybe not even that good anymore. And so just, you know, you're missing so much science. Some people say, well, you know, we can't be taking time out of science classes to talk about creation. We got to talk about creation to talk about science. We're not going to understand the science. We're going to miss the whole point. What was it? A couple of years ago or so, I talked to you about metabolism. You know, just how you break down sugar for energy. And for most books, it's just a series of steps to memorize that make no particular sense because according to evolution 1, things aren't supposed to make sense. According to creation, things make sense. And And you look at metabolism, wow, do they make sense? And praise God, research scientists act like creationists. Even if they pay lip service to evolution, they act like there's a plan and purpose that can be understood, and they expect to see evidence of intelligence in living systems, and they use that to model and choose which experiments to do. And so it really, really works. But another little praise point too. A long time ago, they went through the vestigial organ thing. Man's Darwin and Haeckel, the German Darwin, listed 180 useless leftovers of evolution in the human body. And science proved they were wrong 180 times. Well, then came the junk DNA, and only the media and a few evolutionists, seeking to live on media support, fell for the idea that junk DNA was worthless. The research scientists said, it's there, it's doing something, let's find out what it is. And of course now we know multiple uses for what was once called junk DNA. There just ain't no junk out there except what was brought into the world by man's sin and rejection, of God, and the fall and the curse on the ground. And even then, the evidence of imperfection we see is a fall from perfection, not a build-up from imperfection. Well, we've got time for one more question before coffee and donuts. And again, as always, you all are very patient. Yes.
Even more so is these are the plants that are already there. But yet, when you think that they all came from one cell with the DNA in it,
right,
to build a plant, to build the structure, to build.
Oh yeah.
This is how they operate. But how did they get there?
And that when I was a sophomore in college, that was the big question. One of my teachers, who was a phenomenal, internationally known scientist, and said, that's the key question. All of the DNA is right there in the egg cell. It's all there, but it's expressed differently. In human beings, the cheek lining cells, the CSI swabs take out of you. They can take it out of your liver, take it out of your hair, take it from your eyeball, anywhere. It's the same genetic information. But of course, your cheek lining cells don't look like heart cells or liver cells or bone cells or blood cells. They all have the same genes, but different expressions of that. Some genes are operating, some aren't. And this was, I guess, 60s will be close enough. He was saying, this is the big puzzle in biology, is development. How do you turn genes on and off? How do you know which ones are supposed to work? How do you know which ones work where? And they were doing experiments. I did some of these experiments with frog eggs and with chicken eggs. You take a chicken egg, and you cut a little hole in the shell, look inside, and you see the little future chicken embryo, a little spot on the yolk, and you can dust it with particles of carbon. And it doesn't look like a chicken, it just looks like a little white spot. And then, at a certain time, the carbon particles begin to move. The cells begin to flow. They dive down underneath. Where they dive down underneath, that's where the alimentary canal is going to be. The nerve cord develops above them. The muscle and bone somites develop along the sides. The head raises up, you can describe it in detail. But where's the conductor of the orchestra? The answer is the Lord God, maker of heaven and earth. But he's expressed that in all kinds of ingenious ways. And that, by the way, of course, that's the key to cancer. Cancer's genes not doing what they're supposed to. And that was going to be the key question in the 60s. And do you know they haven't made any progress, hardly at all, on that? I mean, they've done phenomenal things to understand the genetic code, muscle contraction, things like that. They're still wrestling with pretty much the same questions that I was wrestling with as a sophomore in college. It turns out DNA is not the answer. And so the first book I wrote was called DNA, the Key to Life. I would now retitle it Life, the Key to DNA. And so it's all these other things that are telling DNA what to do, when to do it, where to do it. And that's why if you have perfect dinosaur DNA, you can never clone a dinosaur. Because you don't know how to read the DNA. You have to find a dinosaur mother or a suitable egg cell that could read the DNA in order, in sequence, to make the eyes, ears, arms, legs all sprout at the right place and time. And so that is, just phenomenal. Psalm 139. God knew the plan. It was there ahead of time before there were any words in David's mouth. God knew what he would say. He beheld his unformed substance. David started off. is just a little round ball. He was knit together in his mother's womb. Eyes, ears, arms, legs were added in sequence according to the Creator's plan. And that's still the best summary of our scientific knowledge. And what's our response? I will praise thee, for I am fearfully and wonderfully made. And plants would say the same thing, wouldn't they? Okay. Refreshment time, okay? Hey, thanks again.
The only thing I can say is we have one awesome God. That's it.